Welcome back to microbiology. Today we're going to be covering the first half of chapter 4 on eukaryotic cells, what parts are inside, some of their organelles, some subcategories of them, and specifically eukaryotic microorganisms. Eukaryotic organisms that are studied or grouped into microbiology can include protozoa, fungi, algae, and helminths. Protozoa are always unicellular. They're just going to be by themselves. Typically, these are found in aquatic environments, so like pond water, things like euglena or amoeba, things like that. And part of why we care about them in microbiology is because some of them can be parasitic for your patient. Fungi and algae can be unicellular or multicellular. So some examples are fungi. If you were to, go out in the forest and pick a mushroom, of course, that one is multicellular, but we do have things like Saccharomyces cerevisiae, a bread yeast that is unicellular. Helminths are always multicellular, and these are things like tapeworms or flatworms that are also parasites. And the reason that we're talking about them in microbiology is because their eggs are microscopic and unicellular, and the larval forms are microscopic. Obviously, if you have a tapeworm, that can be seen with the naked eye. It's no longer microscopic, but we do talk about it here because of their larval forms. Some key structures to be aware of in the eukaryotic cell are that they have external structures such as appendages and the glycocalyx. That should be kind of similar to what we saw with prokaryotes. They do have boundary structures that look similar, but still have some key differences between prokaryotes. And you'll notice that they have a much wider umbrella for their internal structures. They have membrane-bound organelles, and along with that, they have like the nucleus, the endoplasmic reticulum, a whole slew of very specialized structures. Your book has this image. It's a whole page, and I'm going to zoom in on key aspects of it, but I'd encourage you to go look at it and make sure that you can point out that this is a mitochondria, where's the glycocalyx, and so on. Because again, your first exam is coming, and while it's multiple choice, there probably will be some key questions on the organelles of the cell. Okay, so zooming in on the top part, I'm going to start over here on the left. The cytosol we heard about in the last chapter is going to be that kind of gelatinous, watery component within the cell. We also have cell ribosomes shown here in blue. This is where the proteins would get made. We'd go from RNA to protein, and we talked about this too in chapter 3, how it has a large subunit and a small subunit. So ribosomes are found both in prokaryotes and eukaryotes. They will just be slightly different sizes. So when we talked about the sedimentation rate, whether it's a 70S or an 80S ribosome, that's the main difference. Lysosomes are organelles where we can break down certain molecules. So if there's a worn out organelle or some molecules we need to get rid of, we can put them inside this vesicle with enzymes to help break it down. We have the Golgi apparatus here in yellow. And I've seen in some books, I think it's kind of funny, but it looks like pita bread. And now I can't unsee that. You probably can't either. And so it looks like kind of smushed pita bread here, little pockets, and we can have little vesicles that are passed down along it and are packaged up to be released from the cell. We have the mitochondria. The mitochondria is the powerhouse of the cell. I know you've heard that everywhere, but it's true. Okay, so you have your inner and outer membranes of the mitochondria, and this is where the primary production of ATP, the energy molecule of the cell, is made. We have some cytoskeletal elements like intermediate filaments, microtubules, actin that are going to be used for trafficking molecules or moving things across the cell. Think back to Bio 1 with like dynein and kinesin walking on the cytoskeleton. They're also there to provide structure because the eukaryotic cell has a different boundary than prokaryotes. They rely more on the cytoskeleton than on their cell membranes or cell walls. They do have boundary components though, so they'll have the cell membrane, that's the phospholipid bilayer with some proteins and sterols mixed in. There's also the nucleus, which would likely be the largest organelle if you were looking at it on a microscope, shown in purple. And the nucleus, very spherical, where you would find the DNA wrapped around histones, maybe some transcription factors. It has a membrane, and therefore it has a nuclear pores, kind of these circles that are selectively permeable and let some things in and only some things out, because you don't want your DNA floating out randomly, nor do you want cytoskeletal elements somehow just floating in or enzymes floating in that you didn't want. There's also the nucleolus here, which is a specialized region of the nucleus. And then we have the endoplasmic reticulum down here at the bottom that you'll notice is somewhat continuous with the nuclear membrane. So it kind of pinches off from it, and it's this kind of maze-like organelle. There's the smooth variety over here, and the rough variety, the rough endoplasmic reticulum, is rough because it has ribosomes stuck to the side. We also have the glycocalyx. So this is, again, kind of a slime layer around the outside of a cell, outside the cell membrane, the cell wall. The cell wall is here in green. It's important to note that not all eukaryotes will have a cell wall. So plants would have a cell wall, fungi have a cell wall, but you and I, we're not going to have cell walls. Centrioles are here in yellow. You'll notice that we have some cytoskeletal elements kind of protruding from it. These are used to help organize the chromosomes during cell division. We have chloroplasts here with the thylakoid membranes, the granulose stacks in here, and this would be only in photosynthetic eukaryotes, typically plants, where we would be trying to harvest light energy. And then we have a flagella out here. Not all eukaryotes, again, are going to have that, but some need them for mobility. So we would have some protozoa that might have flagella, as well as some specialized eukaryotic cells, things like sperm, or some of the cells within your intestines may have cilia or some protrusion similar to that for mobility or movement of their external environment. Okay, so we're first going to focus in on appendages for moving. And as I've given you a preview, we have two main types with eukaryotes. We have cilia and flagella. So most eukaryotic microbes, so not the larger organisms, but microbes will have to move in some capacity on their own, and they will use flagella or cilia. Now, it's important that we use the same word, flagella, for both prokaryotes and eukaryotes, but we need to realize that they're not the same structure, though they have the same name. So some key differences are eukaryotic flagella are going to be much larger, much thicker, about 10 times thicker. They're more complex, and they're covered with a cell membrane. So they'll have proteins within the protrusion, and that will be covered up on the outside by the cell membrane. A single flagella is a long cylindrical protein structure made of microtubules. And so you can see over here, it's a 9-2 arrangement. So if you count around the outside, there's 9 sets of microtubules here in blue on the outside, and then 2 on the middle. And then these kind of act as like spokes in the wheel where we can have proteins reaching in towards the middle one and kind of ratcheting around in a circle to help move the flagella back and forth. Cilia have a similar structure, though you're going to typically see many cilia on the surface of the cell, much shorter as opposed to a flagella where typically it would be 1 or very few and much longer. Some cells have cilia to help with feeding and filtering. So that's like my preview within our body. We have cells with cilia in our respiratory tract to help move phlegm down or keep microbes from getting in. You have them in your intestinal tract, things like that. The glycocalyx is that outermost layer of slime basically outside of the cell. Now, your book says that this is the extracellular matrix. And from everything that I've read and ever been taught, that is an oversimplification and not true. And I think you guys can take the full scientific truth here. Okay, so extracellular matrix is true that it's the outer layer connecting cells, but for an extracellular matrix, you would have things like collagen or fibrin, things like that, between like your skin cells and things like that. Okay, this kind of slime layer is very well connected with that individual cell, and therefore it's not truly seen as the extracellular matrix. It is comprised of polysaccharides, similar chemical structure to what you would see in bacteria. And it helps the cell to, again, adhere to surfaces, a lot like the bacteria would. It also helps with signal reception to other cells. So they can send chemical signals through the slime layer to each other. Some eukaryotic microorganisms will have a cell wall. So plants, fungi are the main ones, and they'll have a cell wall to, again, provide more protection, more rigidity. They'll have molecules like glycans mixed in here, glycoproteins that are like sugars and amino acids, and chitin on the outside here. You'll notice that these are very different chemical molecules than what we saw when we were talking about the gram stain with prokaryotes. All cells, whether they're prokaryotic or eukaryotic, should have a cell membrane. And eukaryotes are no different. They have their phospholipid bilayer that's kind of roping off, as you see my image here, roping off between, okay, this is me, this is you, you stay on your side, here's our little boundary. The phospholipid bilayer is going to be selectively permeable. It has polar, non-polar molecules that are all lined up into a bilayer. There are 2 layers of this, and there will be lipids, proteins, and sterols all mixed in. Sometimes we'll say that it's a fluid mosaic model. So with that, we mean that there are those lipids, proteins, sterols, all kind of moving in a fluid. They're not stagnant, or some may be tethered, but most are not, and they can move past each other and get some mixing of these molecules. Sterols or cholesterols can help strengthen the eukaryotic cell membrane and cell wall.